How do different genes cause limb-girdle muscular dystrophy? Have you ever wondered how our genes can impact muscle health? Limb-girdle muscular dystrophy, often called LGMD, is a great example of this connection. It is caused by mutations in various genes that are crucial for producing proteins needed for healthy muscle function. These mutations disrupt the normal operation of muscle cells, leading to progressive muscle weakness and wasting, especially in the muscles around the hips and shoulders. Different genes lead to LGMD by affecting proteins that are vital for muscle cell stability, repair, and contraction. When these proteins are either defective or missing due to gene mutations, muscle fibers become more susceptible to damage during regular activities. Over time, this damage results in muscle degeneration. Currently, more than 30 forms of LGMD have been identified, each linked to mutations in specific genes. These forms are categorized based on the gene involved and how the condition is inherited. The most common inheritance pattern is autosomal recessive LGMD, which accounts for about 90% of cases. In this pattern, a person must inherit two defective copies of a gene, one from each parent. Typically, parents who carry one defective gene do not show symptoms but can pass the gene to their children. A well-known example is the CAPN3 gene, which causes LGMD type 2A, also known as calpainopathy. On the other hand, we have autosomal dominant LGMD, which is less common. In this case, only one defective copy of the gene from one parent is enough to cause the disorder. These forms are sometimes referred to as LGMD1 and can also arise from new mutations that were not present in previous generations. The genes involved in LGMD usually encode proteins that are found in or near the muscle cell membrane. These proteins play a protective role, helping muscle cells withstand injury during contraction and relaxation. When these proteins are abnormal or lacking, the muscle cell membrane loses its protective qualities, making the cells more vulnerable to damage. Some specific proteins affected by these genetic mutations include sarcoglycans, which are essential components of a protein complex that stabilizes the muscle cell membrane. Calpain-3 is another important protein, acting as a muscle-specific enzyme that helps maintain and repair muscle tissue. Dysferlin plays a role in repairing muscle cell membranes, while dystroglycans and other related proteins are crucial for linking muscle cell structures to the surrounding environment. Because different genes influence different proteins, the clinical presentation, severity, and progression of LGMD can vary significantly among individuals. This means that two people with the same form of LGMD may experience different symptoms and rates of progression. In summary, different genes cause limb-girdle muscular dystrophy by producing defective proteins that are essential for muscle cell integrity and function. The specific gene mutated and the inheritance pattern, whether dominant or recessive, determine the subtype of LGMD, its symptoms, and how it progresses. Understanding these genetic causes is vital for accurate diagnosis, genetic counseling, and the development of targeted therapies. If you or someone you know is affected by LGMD, it is always best to consult with healthcare professionals for personalized advice and support.